let's welcome our first emerging scientist, who is Anastasiia Kudriashova, a PhD student with the University of Melbourne and Agriculture Victoria, who applies a wealth of knowledge from her veterinary days in Russia and first-hand experience with challenges of transition cow health in Australia in her search to create breeding values for cows with stronger health and resilience. She will share with us biomarkers for better health as tools for early detection and breeding.
A lot of serious faces. Before we jump into the science, I'd like to paint you a picture. Sorry. Yeah. So before we jump into the science, I'd like to paint you a picture of my dairy farming experience in Australia. It's 4 a.m. in the morning. We are well into the calving. I'm getting the cows in. Automatically checked milk fever, flow pack for milk fever treatment, and moving the cows, and there she is, cast on the side. I treat her, sit her up, mental note to check her after milking. Hopefully she's up. Following the cows, a few red others in front, mainly mastitis cows and cows with uterine infections. There is one skinny heifer. She's obviously crook. I have to call vets. And don't forget to order more drugs. Sounds familiar? Caring for sick cows takes time, energy, and money. And this was a big question for me in the past 15 years. How can we improve dairy cows' health? Transition period is traditionally defined as 3 weeks before and after calving, but its physiological impact may last longer. Up to 75% of all diseases occur during the transition period, and they affect milk production, fertility, and lifespan of dairy cows. If I ask you a question, how can we fix it? You probably will reply, better nutrition and management strategies, select for more resilient animals, and provide with better diagnostic tools that probably work on a herd level. But before we can do it, we need to understand disease biology and whether it is influenced by their genetics. This is where biomarkers come in. Biomarkers are measurable indicators of disease presence and progression. And the aim of my research was identify blood biomarkers and their genetic basis in transition cow diseases. So I collected blood samples from 200 clinically ill cows and 2 healthy controls for each disease case from 15 pasture-based farms, mainly in Gippsland, within 70 days of calving. And I measured this, analyzed these blood samples with multiomics, which means we didn't just test it for one thing, we actually test these samples for everything possible. We genotyped these animals, we checked how genes behave during the disease state, we measured lipids, proteins, and thousands of other molecules called metabolites. And today I will share results with you for my first complete data set, which is lipidomics or FAT. So we identified that lipidomics profile or fats in diseased cows are significantly different from healthy controls. Out of 238 lipids that we measured, 154 had significantly lower concentration in diseased animals. One class stood out, which is lipophosphatidylcholine or LPC lipids. These lipids involved in immune response and inflammation. And these lipids were able to predict disease state with accuracy of 78%, which makes them potential biomarkers for blood disease, for transition diseases. Next, we looked at genetic influence on transition diseases, and we ran we scanned entire genotype to identify genetic regions associated with lipids or fat levels. We identified 35 fats associated with regions on chromosome 11 and 19. Those 20 of those lipids were also significantly different during the disease state, and they were able to predict diseases with accuracy of 72%, which tell us that transition cow diseases have partially influenced by genetics. And take-home messages: using multiomics approach, we are able to discover biomarkers and their genetic architecture of transition cow diseases. Combining all multiomics data like proteins, lipids, and metabolites and gene behavior, we can create complete picture of diseases. And this opens the door for creating better biomarkers that you may be able to use for evaluating your nutrition or management practices. And we can breed for more resilient animals that will be able to handle diseases naturally, transition diseases naturally. Thank you.
Thank you, Anastasiia. Do we have any questions from the audience? Yep, Tom?
Oh, yeah. Do you have any ideas about how the altered lipid profiles relate to disease susceptibility, like in terms of mechanism?
Like, do you want me to go into details? We see significant difference between diseased cows and healthy controls, and there are specific lipids that are different, and mainly they are in lower concentration in sick animals. So it does show us pathways that are involved and show us better like biology behind it. I think if I go too much into details, that might.
Yeah, cool. I might chat later. I've got an interest in the particular genetic variants and the pathways they're involved in, but I'll catch you later.
Yeah.
Excellent. Thanks, Anastasiia.